All right, so today in lieu of class, I want to make this video for you guys. It's voiced over PowerPoint, so I can kind of go back over innate immunity and adaptive immunity briefly. I'm going to kind of rush through it and just cover the highlights. I think this will be really useful for you guys with the exam coming up next Thursday on the 29th. So you can go back and look at this, whenever you want to. If you get confused as you're trying to study, it'll be nice to have this kind of recorded for you. And then after I briefly cover those things again, I'm going to move on and talk a little bit about vaccination and what vaccines are, sort of how they work to generate an artificial type of immunity. So they stimulate the immune system to activate your adaptive immunity. If they are successful, they do. And we'll talk about sort of the vaccine types. And we'll talk about the fact that vaccines are GMOs, which is always kind of a shocker to people when they realize that, hey, this thing is actually a GMO. All right, so remember, the two types of immunity, we've got innate immunity. This is found in all animals, right? Vertebrates and invertebrates. It's part of, well, it's got two kind of categories there where you've got sort of the external barriers. I'm not going to cover those again, but then you've also got the internal defenses where you've got the phagocytic cells and defensive proteins and the inflammatory response. This is very, very different from adaptive immunity. Adaptive immunity is cell-mediated immunity and humoral response, and it involves antibodies and lymphocytes. Okay, B lymphocytes make the antibodies, but then there's also T lymphocytes, which can have a different type response, and they work together and form the adaptive immunity. All right, innate immunity. Here is an example of a defensive protein. The defensive protein in this case is an interferon. These are part of innate immunity. They can be made by virally infected cells. What you're looking at here is host cell number 1, and it is a neighbor to host cell number 2. Maybe they belong to the same tissue, for example. Host cell number 1 is unfortunately infected by this virus. Example, and the virus binds and injects its nucleic acid. And then, of course, this host cell becomes a virus factory. It starts replicating the nucleic acid. It starts making viral proteins. These things assemble and are released from the cell. So this thing is churning out virus. In response to that, though, the cell is not completely helpless. It is helpless in the fact that it is going to become a virus factory, but it's not helpless in the fact that it can actually warn neighboring cells. So it can detect and sense that, hey, I'm a virus factory, and genes are turned on and proteins are made that can actually be secreted from the cell and have an effect on neighboring cells. In this case, the interferon is secreted from the cell. It is then stimulating host cell number 2 to produce some antiviral proteins in preparation and in case the virus tries to bind and infect, right? These antiviral proteins, once made, will prevent replication and infection by this virus. So the host cell number 2 is actually protected against the virus. Host cell number 1, not so much. Host cell number 1's already infected. It's pretty much going to have to be destroyed by a killer T cell or some sort of other phagocytic cell, maybe a natural killer cell. All right, in addition to the defensive proteins, the innate immune response also involves the inflammatory response, or inflammation. This is a major component of innate immunity. It disinfects and cleans infected tissues, and it can limit the spread of infection. Remember, though, this thing comes with the caveat. It's very useful, but if the inflammatory response is too strong due to, you could imagine an overwhelming type bacterial infection, which stimulates an overwhelming amount of histamine release and a large inflammatory response. This can lead to shock, which results in really high fever, and the blood pressure drops and can result in, organ failure and death. So here's the inflammatory response. It's showing you some sort of damage. Here, really what this is showing you is bacteria being poked down into the deep skin layers by the parenteral root, right? So this pen here had bacteria on its surface. It jammed into the finger. Ouch, puncture wound. Bacteria were introduced. And what's going to happen is neighboring cells, damaged cells, cells that sense the bacteria's presence, these guys are going to release some signaling molecules. They're going to be like, hey, immune system, we're going to need some help here. And so these signaling molecules are histamine. I know you've heard of histamine, probably if you've taken allergy medications, because generally these things are antihistamines, and they will block this type of response. But anyway, in the case of the inflammatory response, the histamine is released, and it actually causes an effect on neighboring blood vessels. In this case, a capillary close to the skin surface here is going to be affected. What's going to happen is the blood vessel is going to dilate, so the width of the blood vessel will actually increase. And this is a result of expansion of the cells and expansion of the space between cells. So they become wider and more loosely packed, okay? This actually enables fluid and cells to leave the blood vessel and enter into the neighboring tissue. And you can see these phagocytic cells here are doing that. This rush, this leaving of fluid and cells into the area, this actually causes the swelling. Dilation of the blood vessels causes the redness. Also, swelling is going to put pressure on nerves and cause pain, so there's going to be a few signs and symptoms associated with this. But what's happening when they rush in, these guys are rushing in to do a job. They're going to carry out phagocytosis. They're going to actually engulf these foreign cells, take them inside the phagocyte. They're going to then digest them, break them down into smaller pieces. They will then return to the lymphatic system. So this fluid will drain into the lymphatic vessels, and these cells that have engulfed these bacteria, they will drain into the lymphatic system, where they will then go, and I'll show you later, these phagocytes can activate adaptive immunity. But that's the inflammatory response. All right, so adaptive immunity, let's move on into it. Remember, it's found in vertebrates only. It involves B cells and T cells, right? It involves lymphocytes, B lymphocytes, and T lymphocytes. These are white blood cells, and they actually originate from stem cells in the bone marrow. B cells actually continue developing in the bone marrow. They mature in the bone marrow. That's why we call them a B cell for bone marrow. T cells, on the other hand, will leave the bone marrow, move into the thymus, where they do their final maturation process. So that's why we call them T cells for the thymus. All right. Remember also, B cells participate in what we call the humoral response. They actually release lots of antibodies into the blood, and the blood was one of the 4 humors. And so that's why we call this the humoral immune response, whereas T cells are actually, they actually will directly, killer T cells anyway, will directly engage the invader or the foreign cell or the antigen, whatever it is. And so we call this the cell-mediated immune response, just to differentiate it from the humoral response. All right, remember, B cells and T cells, you've got millions of them. They are, as they mature, they are loaded with receptors of huge variety, okay? Every chemical variety is put into these receptors, every combination of molecule that can interact with potential invaders, okay? But what's nice about this, hopefully, is that these said receptors do not react with your body's own cells. It's really cool how this repertoire is generated and targeted to non-self or foreign cells. Anyway, here's a B cell on the left, T cell on the right, just to show you how cute they are. Let's talk about B cells first. We're going to talk about something with regards to clonal selection. And we're going to talk about a primary immune response and a secondary immune response. Okay, primary being the first time an antigen is introduced or noticed or responded to by the immune system. Secondary would be the second time that antigen, so it would be a second infection, second exposure. All right, so let's start with the primary immune response, and let's talk about clonal selection of B cells. They're showing you 3 different B cells here, but remember, you've got millions, okay? But we're showing 3 for simplification. And think about this as far as the antigen, when it's introduced, it's going to activate and bind to only those receptors that are complementary. So it will only activate and bind to a subset of the lymphocytes, of the millions of lymphocytes in your body. Here's the antigen in this case. It's this little orange diamond, and you can see it does not match the receptors for this B cell, does not match the receptors for this one. It does have the chemical complementarity and binding capacity for this guy in here in the middle. So this antigen will bind to the receptors, and this will actually cause activation, and cloning of this cell. All right, so this cell will get excited. It's going to grow and divide into a clone. All right, so a clone, a group of clones created from activation of this guy. This is the clonal selection part. Now, this group of clones then will split. Some of them will become plasma or effector cells, and they will actually spit out antibodies. Remember, we said something like 2,000 antibodies a second can be spit out by these effector cells. Crazy. And these antibodies then will tag and target. They'll be created, and they are the same receptor and can bind to that antigen which caused activation and formation of these antibodies. Then the other part of this clone will actually be long-lived. So these plasma effector cells, they're going to secrete antibodies just for a period of time. Then their lifespan will be over. They'll die and be gone. But the memory cells can live on for years and years. Okay, and then they will be able to respond fast if the antigen is ever encountered a second time. So let's imagine this antigen is encountered a second time. Here's the memory cells. Remember, they were clones of that original cell that was activated. The antigen comes into the picture again. Second exposure, binding. Now we get activation of all of these memory cells. These guys now activate, grow, and divide. Some of them become plasma cells, effector cells, and secrete tons of antibodies. Some of them then become now a larger clone of memory cells, which can respond if a third time this antigen is introduced. All right, primary response versus secondary response. The primary response is much, much quicker. Is much, much slower than the secondary immune response. Primary response takes more than a week. Secondary response, about a week. Okay? All right, antibodies. What do? We said the job of these effector cells is to make all of these antibodies, but what are they doing? These antibodies can actually promote elimination of this antigen through several different mechanisms here. I'm going to show you them in this illustration. And so what you see here is neutralization. This is when the antibody binding just kind of makes a coat on the outside of the virus or the bacterium that's invading. This actually neutralizes the virus or bacteria. It prevents the virus from being able to bind to receptors on the host cell and enter the host cell. So it's basically all of the receptor spikes on the virus are now blocked. This virus cannot bind to a host cell. Same with the bacteria. He needs to bind and stay attached to the surfaces or the cells of the host in order to not be eliminated or flushed out. And here, with these antibodies surrounding him like this, he can't attach to a surface. Antibodies can also cause agglutination of microbes. They're showing bacteria here being clumped or agglutinating together. This could also be clumps of virus. They're inactivated, they're neutralized in this case. And actually both of these situations, and this one too, will enhance phagocytosis. And this creates a situation where a phagocytic cell can engulf more than one of the bacteria at a time. These antibodies can also be made to some of the toxins produced. Maybe this is an exotoxin produced, like the super antigen produced in toxic shock syndrome, or maybe it's the botulinum toxin produced in botulism, food poisoning, I don't know. But antibodies can be specifically produced to bind to and inactivate and neutralize and cause precipitation of smaller dissolved antigens like toxins. This again this tagging and binding by the antibodies in all of these cases enhances phagocytosis by these phagocytic cells. All right, in addition, you can also have part of the innate immune response activated. This is the complement system. So in some cases, when a foreign cell is bound by antibodies, such as this or this, the complement system is activated. And this is a series of proteins. They're colored green in this case. They actually insert themselves into the membrane of the foreign cell and produce a hole, like a channel. They can do this in multiple places on the surface of the cell, and the cell will then not be able to withstand osmotic pressure. Fluid's going to rush in, he's going to rush in or out, and he's going to lyse and die. All right. What about T cells? We saw what B cells do. They bind to antigens, they get activated, they produce more antibodies and memory cells. And then said antibodies will help promote clearance of the antigen. But then there's also T cells that can get into the picture, okay? Helper T cells can mediate and can actually stimulate B cells further in case they weren't already activated high enough, at a high enough level to the antigen. But then helper T cells can also stimulate killer T cells to come into the picture, and killer T cells can actually bind to and directly engage a foreign cell or an infected cell and kill it. Here's how this works. Helper T cells really only get activated when a phagocytic cell presents the antigen to them. Okay, so this could be a phagocytic cell in the inflammatory response. This could be a phagocytic cell that was activated by, antibodies binding to a target and enhanced phagocytosis. Whatever the case, this phagocytic cell has carried out or has engulfed a microbe and digested that microbe into its components. What will happen then is the components will be put onto a receptor. The phagocytic cell will take the digested components, bind it to a receptor that it has made, and then this receptor gets put on the surface of the phagocytic cell. And the phagocytic cell, as it then returns to the lymphatic system, after engulfing this pathogen, returns to the lymphatic system, circulates through maybe to a lymph node, where it then bumps into a helper T cell and presents the antigen. It is called an antigen-presenting cell because it's basically presenting the antigen to the helper T cell. It says, look, helper T cell, look what I found. All right. The helper T cell is then stimulated by this presentation. In addition, though, the antigen-presenting cell makes some molecules that will actually increase the stimulation and activation of the helper T cell. Both of these, the antigen being presented and the interleukin, the stimulatory molecules made by the antigen-presenting cell, both of these really get the helper T cell excited. The helper T cell then makes molecules to stimulate the B cells. He makes more interleukins, a different kind of interleukin. They're calling this one interleukin 1 and this one 2. So it's just a different form of this interleukin that actually activates a B cell. And it makes an interleukin that will activate the killer T cells. Okay? B cells then will undergo, higher antibody production upon this stimulation. T cells, the killer cytotoxic T cells, do something different. They'll actually get excited, go and bind to the invader, and make a hole in the invader. Let's look at that. They are the only T cells, anyway, that will kill infected cells. They bind to them and destroy them. here's our cytotoxic T cell. He's been excited by his helper T cell activation there. He goes and finds that antigen that matches the antigen that excited the helper T cell. He goes and finds that antigen, or finds a cell infected with a virus that has that antigen there. And this guy will actually bind to that antigen, and this will stimulate him to produce perforin. Perforin is a protein that then is secreted from the T cell, and this thing will actually, it perforates. Think of it that way. It creates a perforin hole, okay? It makes a hole in the infected cell's membrane or in the invading microbe. Maybe this is a bacterial cell. And now it's going to have a hole in the membrane, and then what's going to happen? Lysis. This guy is going to self-destruct there. It's going to lyse and die. All right. Now, of course, both of those types, you know, in all of these cases that we've just looked at with T cells and B cells, this is active immunization. Your body is actively responding to an invading pathogen. But how can we get this response without actually getting sick, right? Maybe we want to prevent infection, and we don't want everybody getting sick. You know, this is nice because when you do get sick, if your body can mount a response like this, or like this, and producing these B cells and producing these antibodies, and this whole sort of clearance here and neutralization and removal of the microbes, that's great when it works, but it's probably pretty rough on a body. It takes a lot of resources and energy, and it's risky because, you know, what if this virus or this bacterium kills you before this actually works, right? So in some cases, we want to get this kind of, we want to get immunity, but we want to get it artificially, right? We don't want to have the infection. We want artificial immunity. There's two ways to do this, okay? You can do active immunization, where we actually administer antigens to a patient. And we administer antigens at the concentration high enough that it will cause the patient to actively mount an immune response. Their body will think they are sick. In other cases, though, that hasn't been possible, and you can actually use passive immunization. This can be, this happens in breastfeeding mothers, for example. Breastfeeding mothers will pass antibodies to the breastfeeding infant there through breast milk. And this will actually allow the infant to acquire immunity. So there's a transfer of antibodies from the immune mother to the developing, you know, the breastfeeding baby. You can also do this, it's also been done, I should say, for example, in cases of, like recent that you may have heard of, is in the cases of Ebola. I don't know if you remember, you know, the American doctor, for example, that was over there treating and helping in the Ebola outbreak. He came back to the U.S. and they found out he was infected with Ebola. They were able to treat him in the hospital. They actually gave him serum. So they took serum from someone who had survived Ebola. Because they had survived Ebola, their blood and their serum contained antibodies to Ebola. They took that serum and took those antibodies and gave it to that doctor. And this gave him passive immunity and helped him overcome that Ebola infection. All right, so let's talk about active immunization, vaccines, vaccination. And then we'll just talk briefly about passive immunization because we've talked about it just a little bit already. All right, so active immunization, usually done with vaccines, and there are a number of different types. Some vaccines actually contain live microbes. These are live vaccines. We generally say they are attenuated or weakened. Attenuated means weakened. It is a weakened live form of the pathogen. These are great when we can do them, because if we can weaken a pathogen and create a live vaccine like this, it is generally going to provide the strongest immune response of the vaccine types. Live microbes will actually produce several antigens, and they will provide a very strong immune response. However, there is risk associated with this because it can result in mild infections. We try to weaken the pathogen so that it doesn't create an infection, but in some cases it does result in mild infections, especially in people that are susceptible or have some sort of weakened state for some reason, where even the attenuated or weakened microbe is strong enough to cause, to overtake their body and cause an infection. Yeah. So in other cases where we feel like the pathogen is just, we can't genetically modify it. I should mention, when we weaken this, we usually go in and remove virulence genes. We find the genes that make this pathogen, pathogenic, give it its virulence, and we actually delete or modify those genes. So it's through genetic modification that we create these. In some cases, we feel like it's too dangerous, right? The pathogen is just too risky. We can't take the risk of a mild infection. In that case, we want to do something different. We want to weaken it further, weaken it to the point that we kill it. In that case, we call this an inactivated or killed vaccine. We generally use an entire microbe or a subunit. These are both very safe compared to live vaccines. A subunit would be where we only take a protein or a portion of the pathogen and put it into the vaccine. This is where a whole agent vaccine is, where you've got the entire organism, the virus or the bacteria, and you've just like heat killed it or killed it with chemicals or something like that. They're both safer, like I said, because you're only using a portion, like a protein from the organism, or it's killed. So you can't really, there's not a risk of a mild infection. However, in this case, you generally don't stimulate a strong enough immune response. The immunity gained from these types of vaccines is not as strong. Because the killed microbes don't produce and don't provide as many antigens, and they don't activate as many B cells and as many T cells. And so what we usually have to do in these weakened vaccines is we have to add something called an adjuvant. We have to add a chemical or a protein or a molecule that we know the body will react to. We add it in addition to the killed agent or to the subunit. And that actually increases the immune response, but it can cause, these adjuvants can cause allergic reactions. All right, you can also use toxoid vaccines. This is done in the case of like botulism or tetanus, where we actually want to maybe not just vaccine against the organism, but we want to vaccinate and provide immunity against the organism's toxins that are produced. So in that case, we might want to produce and isolate large amounts of the toxins. We generally chemically or heat thermally modify the toxins, kind of inactivate the toxins, and then put them into a vaccine, inject them into a host, and that would stimulate B cells to produce antibodies to that toxin. Generally, this requires multiple doses. Very few antigenic determinants are provided by small toxins, okay? And then in some cases, most cases, if you remember, back to when you were a child, I don't know if you've seen the vaccine schedule, I'll show you in a minute. Combination vaccines, you've got several pathogens administered in a single shot, like MMR, measles, mumps, and rubella, right? Exactly. There's 3 in that MMR shot, so that's just one example. Here's sort of giving you an image of how we produce these types of vaccines, and then it also shows you how you might produce a DNA vaccine. All right, so what you're looking at in A, let's talk about each one individually. What you see here in A, you see a virulent pathogen. You see surface antigens, these little triangles and rectangles. That is found on the envelope in this case of this virus. You see the capsid inside with the nucleic acid. All right. In this case, what we're going to make is a live, weakened vaccine. And so what they do in that case is they go in and they look for the virulence genes. Okay, and they're showing the virulence gene in this case in this red, and they're showing, oh, we delete it, remove it. You see now the genome is minus the virulence gene. It's got the blue part, but the red part's been cut out, right? And so this is our pathogen now with the virulence genes removed. We'll take a bunch of these guys, load them into a needle, and give somebody a shot there into the syringe, excuse me. So this is a weakened live vaccine. It says harmless, but remember it does carry some risk, especially in weakened state people. All right, here's example B, where we are just going to use a subunit. Okay, we are going to make a subunit vaccine where we are going to just generate a protein from the surface, an antigenic protein from the surface. This is a bacterial cell in this case, and you can see this little red protein on its surface. It has antigenic properties, and so will stimulate B cells, right, to produce antibodies. But maybe this bacterium is too dangerous, and we really don't want to risk putting a weakened version of this guy into somebody. Maybe that's just too dangerous, too risky. And so what they do is they go into the genome of the bacteria and they find that gene that codes for this protein. And they isolate and purify that gene. Okay? They clone the gene. And they actually probably take that gene and put it into a cell line that they can grow up in the lab. Right? So they introduce this gene for this antigen into this cell. Now when they grow this cell is producing large amounts of this antigenic protein. And so they can grow up large vats of this cell line, and all of the cells in this vat are going to be spitting out this antigenic protein. And then they can purify the antigenic protein, concentrate it, load it into a syringe, inject it into a host to stimulate B cells and T cells. Awesome. All right. In other cases, there's another way they can do this. It's really cool though. Notice all of these involve genetic modification. I love that. It's DNA technology, right? So some of you that are against GMOs might want to rethink this. I don't know. Are you against all GMOs? Maybe you are. That's okay if you are. Just wanted to point that out, right? It's your decision. All right. In example C here, we're going to create a weakened version, attenuated vaccine. It's a live vaccine, but we're not going to use the pathogen. This is a bacterial cell in this case. It's got this antigen on its surface, this triangle. This thing is the antigenic protein. What they might want to do, though, is take this gene, because in some cases this doesn't work as well, right? Because it's a subunit vaccine. It doesn't work as well. So in some cases they have found that this works better. They take the gene for this antigen, isolate it, put it into a virus or cell that will then will then express and generate the antigen on its surface. And they can then take this live cell. Maybe this is a harmless virus. Maybe it's a virus that they have been able to weaken. And they're going to genetically modify this weakened guy to present the antigen on its surface of this pathogen. So they're taking this cell or this virus and making it express and produce this antigen. And now they use this cell or this virus as the vaccine. All right. And then remember, when the final way I want to talk about is a DNA vaccine. We saw this with the example of HIV. HIV is an example of a virus they don't want to take any risks with, right? We don't want to use a weakened, attenuated HIV pathogen. Not going to happen. So what they've tried to do is actually isolate the gene that codes for some of the antigens on the surface of HIV. And they've cloned that gene into a plasmid, and then taken that plasmid and injected the plasmid into a host. So it's the plasmid that is in the vaccine. And then what happens is when this plasmid is injected into your body, it causes some of your body cells to produce the pathogen's antigen and stimulate an immune response. Wow. So far, this hasn't worked very well. It hasn't been long-lived enough. They've had trouble getting the body cells to produce enough antigen to stimulate an immune response, but they're working on it. All right, this is just showing you, how many vaccines has a typical person had? This is the CDC recommended immunization schedule. This is old. I probably should update this, but it's from 2011. And it shows you from birth all the way to old, right? Greater than 65. Most of vaccines are done in childhood. You'll notice most of them have multiple doses. Here's hepatitis B, dose 1, dose 2, dose 3. And then there might be a need for boosters later. Rotavirus, you get a couple of those. DTP, diphtheria, tetanus, pertussis. You get numerous DPT shots. Human papillomavirus, this is a newer one. There's an HPV vaccine now. This is done at adolescent years. Typically series of 3. You can see some of the others. Influenza, annually. It doesn't provide a long-lasting immunity because influenza mutates quickly. MMR, measles, mumps, and rubella. These are just a few examples. I don't want you to memorize this at all. I just want you to see that you've probably gotten a lot of vaccines and probably will get a few more in your lifetime. All right. Now, active immunization using these vaccines, it really is the best way to generate an immune response. However, it can result in mild disease in some people. Most people don't feel too well after they've had a vaccine, even if they don't get a form of the mild disease, because when your immune system mounts that response, which is good, it typically generates some of the symptoms and signs of the illness itself. So sometimes people will think they have the illness, but actually it's their immune response that's responsible for some of those symptoms. But in some cases, they do actually get a mild form of the illness. You could also, it does come with a risk of anaphylactic shock if your immune system responds too strongly. If the inflammatory response is activated for some reason, and it's activated in a strong way, blood pressure can drop, shock can ensue, death. Then there's also the allegations out there, and I know you've probably heard of these. There are certain people, there's a lot of people actually that are anti-vaxxers, for example, and they don't believe in vaccination. They say that vaccine causes autism, diabetes, asthma, and other things. If you are somebody that's on the kind of on the fence about this and not sure what to think, I really do encourage you to look into this. Look at the research, not just some of the, secondhand articles published out there. Look at the actual research, because if you look at that, you'll realize there really is not a connection between vaccines and autism, for example. It's just not there. All right, passive immunotherapy. Remember that? We talked about that briefly. This is when you give, this could be done by breastfeeding mothers, or if someone has survived an illness recently, their bloodstream will probably be full of antibodies. And so you can actually take the blood and isolate the serum containing the antibodies. They call this antiserum because it has the antibodies preformed in that individual's body while they were infected. And since they've healed and they're cured from it, they've got lots of these antibodies. So then you can take that antiserum and inject it into someone that is currently sick with this infection, and it can help cure them. There's several limitations to this, though. It can trigger allergic reactions because antiserum from one person's body may contain foreign molecules that trigger the immune response to act against that serum in certain hosts. That serum could be contaminated with other illness that maybe wasn't purified completely away, other viruses. So there is a risk of contamination. And then the antibodies in, the antiserum, they don't last that long, okay? They provide protection for just a brief period of time. Now, we're not going to talk about the bullet ant. I should have hid that. Limitations of that can be overcome by production of these things called hybridomas. This is pretty interesting, I think. To create a hybridoma, what you are creating is a cell line that will make antibodies to a certain pathogen. You could do this with Ebola. You could take Ebola, inject Ebola into a mouse. Poor mouse. And then let this mouse's body act and activate and produce antibodies to the Ebola as he tries to fight off this Ebola. All right. And then what you can do is you can take these B cells, these plasma effector cells that are secreting these antibodies to the Ebola, and you can isolate them. Okay, that's what they're showing here. And this is the interesting part. You would take this B cell and fuse it with a cancer cell that you've been growing in the lab. So they're showing a myeloma. This is a cancer cell line grown in the lab in culture. They take the myeloma cell and they take the B cell and they fuse them. This produces, because cancer cells never die. We know that plasma effector cells are short-lived. If we fuse this short-lived cell with this long-lived cell, you create basically a cancer B cell. You create this long-lived B cell that will live for a very long time and produce large amounts of antibodies. And you can grow this hybridoma, this cancerous B cell, in the lab in large amounts in culture, and you can allow it to produce the antibodies to this, whatever you injected in the mouse. In our case, it was Ebola. Could have been something else, though. And now we've got antibodies we can isolate. And it's not from a host. It's coming in a cleaner, less chance for contamination, and we can make as many of them as we need. Okay, pretty cool idea. This is just showing you a graph of how sort of active immunization would look. This is showing like your first initial shot vaccine. Your body's gonna, and it's showing antibody, so it's time versus antibody concentration they're measuring. And at the initial vaccine, over time, you're going to get some antibodies produced, and then it's going to sort of level off. And then you're going to get a booster, which is going to shoot it up. You're going to produce more antibodies, and it's going to level off. And you get another booster, and it shoots up even more. And so you get a long-lived production of a large amount of antibodies. Where this is showing passive immunotherapy, if someone was sick with something and they were injected with antibodies that were preformed either by a hybridoma or in someone else's body, like in their antiserum, and I got an injection of those antibodies, it's going to give me a high amount of protection at the time of injection, but it's going to slowly decline over time.